Welcome back to the Talking Ketamine podcast. This is episode 39, The Neuroplastic Revolution.
It's good to be back. Today we are marking a special anniversary. It has been over 25 years since the first study showed ketamine could stop depression in hours, not weeks. We have a retrospective paper from 2026 that tells the story of how that one discovery completely broke the old model of mental health.
It is. It's wild to think about. Happy anniversary to the revolution, I guess.
I think so, yeah.
It's January 2026, and looking back at this quarter century of change, honestly, reading this paper felt a little like reading a mystery novel where you finally figure out who the killer was all along.
That's a very good comparison. Because for a long, long time, psychiatry was essentially investigating the wrong suspect.
Right.
Or, maybe they were focused on the weapon, but not the actual person using it.
And that suspect was serotonin. I really want to start there because I feel like we need to vindicate the millions of people who lived through the Prozac Nation era. If you walked into a doctor's office in, say, the 90s, even the early 2020s, and said you felt terrible, you got a very specific story. They told you, hey, your brain is a chemical soup, and you're running low on one ingredient.
The famous chemical imbalance theory, which is arguably the most successful medical marketing campaign in history.
Exactly. It was the fill the tank model. My brain's a car, the serotonin tank is empty, and this pill is just, pouring more fuel in.
Yeah.
Simple, logical. It was everywhere.
It was.
But this paper basically says that for 50 years, that model was it a lie? Or just really bad science?
I don't think it was malicious, so maybe lie is too strong. It was just woefully incomplete. We call this the Monoamine Dogma.
The Monoamine Dogma.
Yeah, from the 1950s all the way through the 90s, the entire field was obsessed with these monoamine, serotonin, norepinephrine, dopamine. But the logic they used was, it was circular. And that's what kept everyone trapped.
How was it circular?
Well, back in the 50s, we accidentally found some drugs like tricyclics and later SSRIs that boosted these chemicals. And because some people felt better, we sort of worked backward from there. The thinking was, okay, if the drug boosts serotonin and the patient gets better, then the lack of serotonin must have been the cause in the first place.
That feels, a bit like a logical leap.
It is. It's like saying aspirin cures my headache, therefore all headaches are caused by a lack of aspirin.
Okay, that actually makes perfect sense when you put it that way. But people did get better, right? Millions of people take these drugs.
So the theory had to have some legs. Why did every doctor repeat it if it was that shaky?
It had legs, but it definitely had a limp. It worked enough to keep the theory alive, but it failed enough that it should have suggested we were missing the big picture. And this is where that glitch in the matrix starts to appear. It's fundamentally a math problem that scientists just kind of ignored for decades.
I love a good math problem in biology. Lay it out for me.
Okay, so when you take an SSRI, we know from just basic pharmacology that your serotonin levels in the synapse, that gap between your neurons, they shoot up almost instantly.
Within hours, maybe even minutes. Your tank is, for all intents and purposes, full.
Okay, so functionally, the chemical imbalance is fixed on day one.
Chemically, yes. But do you feel better in a few hours?
No, definitely not. Every doctor tells you it takes time to kick in, usually 4 to 6 weeks, sometimes longer.
Exactly. So there's the paradox. If the problem is just a low tank and I fill that tank on day one, why does it take 30 days for the engine to actually start?
That is a massive lag.
It's a huge latency problem. If you were dehydrated and I gave you water, you wouldn't wait a month to feel less thirsty.
Right.
If you have an infection and you take an antibiotic, the bacteria start dying pretty much immediately. This lag suggested the serotonin boost wasn't the cure itself. It was just the first domino in a very, very long, slow chain of events.
So we were just waiting for the brain to adapt to the drug instead of the drug fixing the problem directly. And the paper mentions the STARD trials, which sounds like a sci-fi movie, but I guess it was a pretty damning reality check for this theory.
Oh, it was the largest antidepressant study ever done. And the results were humbling, to say the least. They found about a third of patients didn't respond at all.
A third?
Even after trying multiple different monoamine drugs. And for those who did respond, the effects often faded. It proved that simply filling the tank just wasn't working for a huge chunk of the population.
And then you have the tryptophan depletion paradox, which I found fascinating because it really tests the core logic.
It does. Tryptophan is the amino acid your body uses to build serotonin. So if the low tank theory were true, if I took a healthy person and just drained their tryptophan, lowered their serotonin artificially, they should instantly plummet into a deep depression.
And did they?
No. Not at all. They were fine. Maybe a little grumpy, a bit irritable, but not clinically depressed. So by the late 90s, the smartest people in the room knew the chemical imbalance story was too simplistic, but they didn't have a better story to replace it with. They were stuck, just waiting for a spark.
And according to this retrospective, that spark came from a very, very unlikely place.
Right.
A club drug.
The accidental revolution.
Ah.
Yeah, you have to remember the context of 1999. Ketamine was either an anesthetic used in surgery, or it was Special K on the dance floor. It was definitely not considered a precision mental health tool.
It sounds almost reckless, like trying to cure a headache with tequila. Who looks at a dissociative party drug and thinks, yes, this is the future of psychiatry?
Well, desperation breeds innovation.
Mm-hmm.
A team of researchers, Berman and his colleagues, they decided to test the glutamate system. They suspected maybe the monoamines were just a dead end. So they took 7 patients with major depression people who had failed other treatments, and they gave them a tiny sub-anesthetic dose of ketamine.
And this is the moment the history books rewrite themselves.
This is it. Because of the speed. Remember that 6-week wait for SSRIs?
Mm-hmm.
In this study, the patients reported significant relief in 240 minutes.
4 hours.
4 hours. That's
Yeah.
not just an improvement. That's a different species of treatment. It's the difference between mailing a letter and sending an email. Precisely. That speed just shattered the monoamine dogma. Because 1, ketamine doesn't even target serotonin. It targets glutamate, specifically the NMDA receptor.
Right.
But more importantly, because it worked in hours, it proved the brain could be fixed quickly. It wasn't about slowly building up some chemical reservoir. It had to be doing something else, something totally different.
So if it wasn't filling the tank, what on earth was it doing? Because if I can fix a machine in 4 hours, it usually takes 6 weeks to repair. I'm not just topping off fluids. I'm probably flipping a switch or, like, fixing a wire.
You are hitting on the key metaphor shift. This is where we move from chemistry to structure, from soup to wiring. The paper calls this the plasticity revolution.
Neuroplasticity is a word we hear all the time now, usually to sell brain games or supplements. But let's unpack what it actually means for depression, because I think people imagine the brain just getting...
It's a bit more precise than that. Think of it this way. In the early 2000s, as we look closer at the brains of depressed patients, and also animals under stress, we started seeing physical damage. Chronic stress acts like a drought in the brain.
The drought.
Yes. If you look at the hippocampus, your memory center with the prefrontal cortex, the neurons actually start to wither. The branches, which we call dendrites, they retract. The leaves, which are the dendritic spines where all the communication happens, they literally drop off.
So the brain is physically disconnecting itself.
It is physically atrophying. The hardware is degrading. It's losing the ability to communicate with itself. The complex web of connections that makes you you,
Yeah.
it's thinning out.
That's terrifying, but it also explains so much. I mean, depression feels like a shutdown. You feel disconnected. And it turns out you are disconnected. It's not just a feeling, it's a structural reality.
Exactly. And this is where ketamine comes in as the antidote. If stress is a drought, ketamine is like a rapid-release fertilizer. It triggers a massive surge of a protein called BDNF, brain-derived neurotrophic factor.
Right, we've called this Miracle-Gro for the brain before on the show.
And it's a very accurate analogy. And the tech caught up to the theory. The paper highlights how we started using 2-photon microscopy to actually watch this happen.
You can see it.
You can literally see the dendritic spines popping back out on the neurons hours after a dose. It's like watching a time-lapse video of a forest blooming in spring, but it's all happening inside your head.
That is such a powerful image. And it clarifies why SSRIs were so slow. The paper says SSRIs do eventually boost BDNF.
They do.
Yeah.
But they take the scenic route. They have to kind of nag the brain for weeks with these chemical signals before the brain finally agrees to release the fertilizer. Ketamine kicks down the door and dumps the fertilizer on the lawn immediately.
So the problem was never just the fluid in the tank. The problem was that the engine parts were rusty and disconnected. Ketamine reconnects them.
That's the psychoplastogen model. We're not just tweaking mood. We are inducing structural repair. But, and this is where this 2026 paper gets really interesting, repairing a few synapses, is microscopic. How does that translate to me? How does growing one dendritic spine stop me from feeling like a total failure.
Right. We have to connect the micro wiring to the macro experience. The paper talks about multi-scale plasticity. I feel like this is where we can sometimes lose people in the science, but it's crucial. We have to bridge that gap between the molecule and the mind.
Exactly. So let's start at the trigger. We mentioned the NMDA receptor.
The gatekeeper.
Right. In depression, there's a specific brain area called the lateral habenula. I like to call it the disappointment center.
That sounds like a terrible place to hang out.
It is. In a healthy brain, it tells you when you've made a mistake so you can learn from it. But in depression, the lateral habenula is firing in these bursts. It's just screaming constantly. It's inhibiting your reward centers, basically telling you, don't bother, it's not worth it, you suck.
So it's a biological killjoy. It's physically suppressing your ability to feel good.
Correct. Now, here's the mechanism. Ketamine blocks the NMDA receptors right there in that area and stops that burst firing. It effectively shuts up the disappointment center. So step one.
Okay.
But that blockade sets off a cascade. It activates a pathway called mTOR.
I've seen mTOR described as a protein, but what's its job?
Think of mTOR as the master construction foreman. In a depressed brain, that foreman is asleep on the job. Ketamine wakes him up loudly and he goes, "Okay, crew, start building." And that's when the proteins for new synapses start getting manufactured.
So that's the chemical switch. Stop the disappointment signal, start the construction signal. But how does that change the loop in my head, the rumination? Because that's the part of depression that really tortures people, the thoughts.
And that brings us to the macro level, the network revolution. By around 2015, we stopped looking at just brain regions and started looking at circuits. The big villain in depression became the default mode network, or DMN.
I know the DMN, that's the me network. It's what's active when I'm daydreaming or worrying about the future or replaying an argument in the shower.
Exactly. It's self-referential processing. In a healthy brain, the DMN turns on and off. It's flexible. You focus on a task, the DMN quiets down. In depression, it gets stuck in the on position.
Stuck on.
It becomes hyper-connected. And that is the rumination loop, the inner critic that will not shut up. It's a closed loop of negative self-referential thought that just dominates your attention.
It's like a browser tab that's frozen and playing loud music, and you just can't close it no matter what else you try to do on the computer.
That is a perfect description. And ketamine is the reboot, because it disrupts that hyperconnectivity. It temporarily scrambles the signal, allowing the DMN to reset and normalize its connection with the emotional centers of the brain. It breaks the loop.
I love the reboot analogy because it explains the relief that patients feel. It's not just that they're happier instantly, it's that the noise finally stops. The system creates space.
Space is the perfect word. It creates a window of plasticity where the person can step outside their suffering and start to build new patterns. It doesn't force you to be happy, but it gives you the capacity to be happy again.
And this brings us to where we are today, in 2026. The paper argues we've moved into this era of precision psychiatry. That sounds a little like marketing jargon. What does it actually mean for a patient walking into a clinic today versus, say, 10 years ago?
It means the end of the guessing game. For decades, psychiatry was just trial and error. Try this Prozac. No. Okay, try this Zoloft. No. Well, maybe talk therapy.
Throwing darts in the dark and hoping you hit the board, and meanwhile the patient is suffering for months or even years.
Right. But now, because we understand the hardware versus the software, we're using biomarkers. We can do scans or blood tests to see. Is your depression driven primarily by inflammation? Is it a glutamate issue? Is it a network connectivity issue in your DMN?
So if my DMN is stuck, maybe I need a neuromodulator like ketamine or TMS. If it's inflammation, maybe I need an immunomodulator. We can actually treat the cause, not just the symptom.
Exactly. The paper makes the point that future therapeutics and current ones, here in 2026, are judged by their ability to restore flexible, resilient brain circuitry.
Resilience seems to be the key word now. We aren't just trying to numb people to the world.
No. The goal isn't to sedate you so you don't feel pain. Life is painful. The goal is to make the brain capable of bouncing back from stress again. We're trying to build a stronger engine, not just put a muffler on it. We want a brain that can take a hit and keep on functioning.
It's truly amazing to think this entire paradigm shift started with a drug that people just dismissed as a horse tranquilizer.
It really is. Ketamine was this sort of molecular probe that accidentally revealed the structural nature of our minds. It forced us to look at the wiring, not just the fluids. It changed the entire question, which finally allowed us to find the right answer.
And that shift is why we're celebrating today, because it gave us a map.
A blueprint.
So we've moved from filling the tank to rewiring the engine. It turns out the problem wasn't just running on empty, it was a broken connection.
Exactly. The last 25 years showed us the problem isn't just chemical, it's structural. And ketamine gave us the blueprint to fix it.
Here's to the next 25 years of discovery. Thanks for listening to the Talking Ketamine podcast. We'll catch you on the next one.